Accident of funding, NIMH stayed open, whereas most other government offices are closed. I'm sure for some very good reasons.
Not.
But, yeah, not. And this has not affected Betsy, so that's our good fortune. Betsy is now the chief of the Laboratory of Neuropsychology and the chief of the section on neurobiology of learning and memory at NIMH. She's been there for quite a long time. I think she started there about the same time Kolb started at Lethbridge, approximately, late 70s.
I started here in '76. She looks so much younger
78.
than you.
I think you started 78 at NIMH?
Or 79. I think
79?
I didn't get my degree till later. Yeah.
Yeah. And she has been the author of a great many influential papers. In my history, I think the first one I was aware of was on cognitive processes in monkeys with Mort Mishkin. And in fact, one of those papers, which were classic papers, was on cross-modal associations in monkeys with amygdala damage. Rob McDonald's very first experiment was to try to replicate that finding in rats. And he got the result exactly wrong. He found a hippocampal effect and not an amygdala effect in cross-modal associations. She has continued to work on cognition in macaques and quite recently published a book on evolution of memory systems, which I can recommend even though I haven't read it yet. My apologies.
It's okay.
I've heard quite a bit about it. She has won many honors and awards. I'll just talk about a very small number of them. She won a Distinguished Scholar Award in 2018 from the Social and Affective Neuroscience Society. She's a fellow of APS, Society for Experimental Psychologists, and APA, and she's won more awards in-house at NIMH than I can list throughout her career. The work she's done with monkey has included some very nice work on functions of hippocampus, amygdala, ventral striatum, mostly in learning and memory. And then more recently, she's been focusing on prefrontal cortex in decision making. And that's the topic of today's lecture. That's it.
Okay. Thank you, Rob. Thank you. Thank you. So this is doing a bit of testing here. That sounds good. My voice doesn't carry very well, so really if you don't hear me in the back, please let me know. I'm really pleased to be here. I'm really honored to be invited to deliver the Harley Hotchkiss Memorial Lecture, one of your Harley Hotchkiss Memorial Lectures here at the Canadian Centre for Behavioral Neuroscience. This is my first visit to the center, so I'm very excited, and I've already had a good time talking to people about their work, and I'm looking forward to hearing more. So let's hear it for behavioral neuroscience. So this morning I'll be discussing some of our work on the neural substrates and the frontal cortex contributions to learning and decision making. This is our subject, a macaque monkey, and this photo resonates with the topic of my talk, which is how do we identify and select targets or goals for action. And the work I'll talk about today is derived from a research program that makes use of selective cerebral lesions and disconnections, some temporary inactivations, combined with behavioral analyses, really just to try to understand how information about objects and events is stored in the brain and how this stored information, especially information about objects, is linked to reward and emotion and to action. On the bottom here, some drawings of a human brain on the left, macaque brain, and then a lateral view of the rodent brain. Rostral is to the left. And you can see exploded in the top are these sort of exploded views of the orbital cortex. the cortex is color-coded. So the yellow cortex is allocortex that has homologs in all vertebrates. The green stuff is a granular frontal cortex, which is common to all mammals. And the blue stuff is granular and dysgranular frontal cortex that's specific to primates. So part of the reason I study monkeys is because these areas, all this stuff in blue, I'll just read it from the slide, is prefrontal cortex that has homologs in macaques and humans that other mammals lack. So really, there are just 2 points here. One is that, according to Price and colleagues, many of the same cytoarchitectonic areas have been identified in macaques and humans. So whatever we might learn about the macaque brain may well inform the human situation. And this is a cladogram at the bottom here of Euarchontoglires. So Euarchontoglires is a lineage starting with the last common ancestor back here of rodents and primates. And this just shows some of the parts of the brain I'll be talking about today. One of those is the granular orbital frontal cortex, which emerged in early primates. And another, so labeled here OFC. And another is the ventrolateral prefrontal cortex, or VLPFC, that arose in anthropoid primates a bit later, maybe 35 million years ago. And of course, those are the common ancestors of New World monkeys, Old World monkeys, apes, and humans, so us. So all these guys are our ancestors. It's rare that we're instructed about or explicitly told about a good choice or bad choice. Of course, in practice, what we have to do is learn about or otherwise infer the best course of action. And the work I'll talk about today is really just getting at one tiny area of decision making, which has to do with representations of value and changes in those representations of value. So here's our weather forecast for today. First I'll be talking about a reversal of fortune, which is really what the orbital frontal cortex does not do, and that will become apparent in just a minute. The bulk of my talk really has to do with mechanisms of valuation updating, and for the purpose of this talk, I'm using the word valuation simply as a shorthand for representations of value. Then I'll spend just a minute trying to put those findings in some sort of evolutionary perspective. And then finally, if there's time, just a few minutes about what we're doing now. So there are many ideas about orbital frontal cortex function. I'll just go through a handful of them, and we'll try to see how the evidence that we've collected holds up. One of those ideas is that the orbital frontal cortex is important for behavioral inhibition and emotion regulation, for that matter. It's next. And these ideas have a long history and a long tradition in human neuropsychology and have mirrored findings in macaques. Another idea that comes more from learning theory is that orbital frontal cortex is important for outcome valuation, literally for something like representing the properties and value of rewards. And this is sort of a very different idea from a different literature. And then also more recently, an idea has come up that the orbital frontal cortex is important for credit assignment, literally for understanding the relationship and learning about the relationship between a particular outcome that you might have gotten, like a food or juice, a reward outcome, and the choice that produced it. And so all of these, well, as I said before, these first two ideas really are the foundational ideas about orbital frontal cortex. And they come from a history of human patients with orbital frontal cortex damage being impaired, often especially in reversal learning, stimulus reversal learning. Also, for the emotion regulation part, showing affect that's inappropriate for the context in which they're in, or sometimes blunted affect. And as I said, there are parallels in animal work. And in all these tasks, there are tasks that have been used to try to index those behaviors or those processes. And first and foremost is, of course, stimulus reversal learning. This really involves asking humans or animals to attach stimulus information to reward information, and then changing the circumstances or the contingencies. But the value of those tasks is they can be applied in humans and across several animal species. In macaques, we've used the snake test as a test of emotion regulation. And that's because when we expose monkeys to a fake snake, we can actually pit approach responses to obtain food against the defensive responses engendered by the snake. And so it's arguably a test of emotion regulation, at least a crude one. Outcome valuation can be studied by a devaluation task, which I'll describe a bit later. And credit assignment is usually estimated with probabilistic learning tasks. And again, I'll describe that a bit later. So we're going to go through these ideas on the talk. So I've been studying orbital frontal cortex for many years now, more than I care to admit. And as you can see, some of the findings come from long ago, like around 2004 and 2013. In the next few minutes, I'll actually be comparing data gathered from monkeys that sustained excitotoxic lesions of the orbital cortex, which are shown sort of slightly here in T2 weighted MRI scans, or aspiration lesions of the orbital cortex, shown here in these structural MR scans. And I think you can see the gray matter of the orbital cortex is missing here. And in here, this white hypersignal shows the locations of injections of excitotoxins. And the white really just shows the location of edema, which maps on pretty faithfully to the location of the lesion later. So, as I said, we've been studying orbital cortex for a long time, and only relatively recently did we evaluate the effects of excitotoxic lesions. And that's just because it was technically difficult to achieve that. Our first results were actually looking at the effects of subtotal lesions of the orbital cortex, and the results really did not make sense to us. So we realized right away we had to step back and simply evaluate the effects of complete lesions of the orbital cortex made with excitotoxins. So what I'm going to show you in the next few slides literally are data from monkeys and those data were collected about 10 years apart. So you may say, oh God, this is crazy, you know, how can you do that? Well, I think we feel comfortable doing it because, first of all, the boundaries of the lesion are exactly the same. So the lesion of orbital cortex is just sort of a standard lesion, including areas 11, 13, and 14. The boundaries of the lesion on this ventral view of the macaque brain extend from the lateral orbital cortex here, here, onto the medial surface, to here, and here, and here. And in addition, the monkeys were trained on batteries of tasks that were really the same battery of tasks using the same procedures. So I'm going to show you this data. One of the tasks in the battery is object reward reversal learning. And this is just to remind me to tell you that it involved a single pair of objects. This is really trivial. Monkeys learn to approach and displace the rewarded object and to avoid the unrewarded object. When they've learned that over the course of a couple days, then we reverse the stimulus reward contingencies. The animals, again, have to essentially relearn, and they learn to a criterion. And then when they've done that, we reverse the stimulus reward contingencies again, and so on for several serial reversals. And so that's what you'll see on the next slide. So here on the ordinate are errors to criterion. Down here is acquisition, and all these numbers. It means every time the monkeys relearned the discrimination, then the contingencies are reversed, and they go on to the next reversal, and so on. So shown here are the errors scored in acquisition. All the monkeys do that very efficiently. This is a really trivial task for monkeys to acquire this discrimination. But as soon as you introduce a reversal, the monkeys score more errors. So our control groups are shown here in the gray curves. These are unoperated controls. I won't be saying much about ends of groups. These are all, ends are standard sort of 4. If I say something significant, it's either an analysis variance or a non-parametric. Anyway, I'm not going to be saying much about ends. So here are controls, and you can see the classic result of aspiration lesions of the orbital cortex is these monkeys are scoring huge numbers of errors. They're finding it very difficult to reverse. And whereas our intact monkeys are actually learning more efficiently across the reversals, they're developing a reversal learning set, these monkeys with OFC lesions do not. So this is really the classic result, this profound impairment in stimulus reversal learning. We've known about it for decades. This just happens to be the data from my own lab, but we've known about it since the time of Charlie Butter in the 60s. When we tested our monkeys with excited toxic lesions, this is the result we got. These animals are simply not impaired on this task. We didn't really want this result, we didn't believe it, so we added more monkeys to the group. We certainly didn't know what to make of it because errors to criterion are arguably not the most sensitive measure. We took measures from reinforcement learning. So for example, we looked at the influence of strings of errors or strings of correct responses on the subsequent choice. And again, there was no hint of an impairment. So these data strongly suggest that the aspiration lesions of orbital cortex are having their effect because of disruption of fibers of passage, that is, disruption of fibers passing nearby or through the orbital cortex en route to somewhere else. And to test that idea, we took some of the monkeys from the main experiment and gave them these narrow strip lesions of orbital cortex. So the idea was these were narrow aspiration lesions in the caudalmost point of our orbital cortex, granular orbital cortex region. And those animals were retested on the stimulus reversal task. And that lesion sort of reinstated the impairment, despite the fact that now this is the animal's second time around on this task. So these data too, and so these animals are impaired, and they're as impaired as the animals with complete aspiration lesions of the orbital cortex, even though most of the orbital cortex is actually intact. So these data really strongly support the idea that the aspiration lesions are having their effect, at least on some tasks, because of a disruption of fibers of passage. Whoa, okay, that's interesting. And then the question is, okay, what fibers of passage? Well, the main fiber bundle that's sort of connecting temporal lobe and frontal lobe, but is actually connecting many, many more things than that, is the uncinate fascicle. And it's really a sheet of fibers coming up from the back of the brain into the frontal lobe that's spreading out over the orbital cortex. And the main fibers going through there are lots of projections from the medial temporal lobe to the prefrontal cortex. So that includes entorhinal cortex, perironal cortex, perihippocampal cortex, many of those fibers ending up in the medial frontal areas. It also contains lots of projections from visual area TE. This is lateral temporal cortex. And many of those projections are ending up out here in the ventrolateral prefrontal cortex. There are also many intra-prefrontal connections. So I'm showing this figure from Lehman et al. From Suzanne Haber's lab, simply because it's a nice diagram of the trajectory of fibers arising from these different injection sites in the orbital cortex. And you can see how the uncinate fascicle, labeled UF, is sort of hugging the gray matter of the orbital surface. So there are many, many fibers going through that region of the brain. We're actually currently trying to conduct, or we are conducting, traditional track tracing experiments trying to understand what fibers are disrupted by an aspiration lesion of orbital cortex. I don't have an answer for you today, but I think it's clear the bottom line is that damage to white matter coursing nearby or through the orbital cortex after aspiration lesions is causing deficits in reversal learning. So of those ideas we started with, these data have led my colleagues and I to reject the idea that the overarching function of the orbital cortex is in behavioral inhibition and cognitive flexibility. I know that's putting making sort of a grand statement on a small amount of data. And there are nuances about reversal learning that we might want to talk about later, having to do with what's happening in marmoset monkeys or rodents or other animals. But in macaques, it just is strikingly clear that orbital cortex is not contributing to that function. I don't have time to talk about emotion regulation and show you those data. Interestingly, our monkeys with excitotoxic lesions of the orbital cortex are also totally unaffected on the snake test. So there is no emotional blunting. Their defensive responses are just fine. So again, this just doesn't seem to be quite what the orbital frontal cortex is doing. And I have to add a slight caveat. We have some recent data that arise from looking at the effects of subtotal lesions of the orbital cortex, which is bringing this into question. So I'll probably replace that and just make it a question mark, but that's not part of what I'll be talking about today. And that brings us to this updating outcome valuations story, which really brings me to the main part of my talk. So we'll be looking at neuromechanisms of valuation updating, and part of the main way to do that in animals is with a devaluation task. It's just jargon, but like the reversal learning task, it requires monkeys to discriminate objects. Monkeys' visual systems are very much like your own. That's a really trivial problem for them. But unlike reversal learning, it requires animals to link those objects with food reward value. And I'm saying that, and I know that, because what we're asking the animals to do is learn about associations between specific objects and two different foods, some associated with one food and some with another. And then we're going to manipulate the value of that food and see how it influences monkeys' choices of objects.
Betsy, when you use that term, is incentive the same thing? Incentive value?
I think
Or
so,
do you like
yes.
incentive value, or?
I know there's so much jargon out there that it is hard to know. So I just say literally food value because I feel like I know what it is. But yes, I think people would use the term incentive learning and incentive value, yes. So we use the procedure of selective satiation to manipulate food value. And these frames from the film Cool Hand Luke really capture that nicely. So this is the famous hard-boiled egg eating contest from the film Cool Hand Luke with the late Paul Newman. And you can see that at the beginning of the hard-boiled egg eating contest, Paul Newman is happy to eat eggs. He likes hard-boiled eggs. But by the end of this hard-boiled egg eating contest, he simply cannot countenance eating another egg. So you've probably all had your own run-ins with selective satiation. Maybe need this slide, but as in theory, the idea is that the food, or more accurately, the sensory properties of the food are linked to some emotional tag that's literally producing some distaste of that food or anticipation of that food. So in our devaluation task, there are two phases. First, the monkeys learn about a large number of objects and their associated food rewards, and the vehicle for this is a discrimination learning task. So literally, monkeys learn this very nicely. Some objects are associated with one type of food, say peanut. Others are associated with a different food, say M&Ms. And then in the context of discrimination learning, some of these are simply negative objects. They're never rewarded. And when the monkeys have learned these discriminations, we bring them to a series of probe tests. And first we run a baseline test. And we set aside the negative objects, and now for the first time, monkeys are faced with making choices between two rewarded objects, one which had been associated with food 1 and the other with food 2. And in these baseline choices, we simply record the monkey's choices. There is no wrong answer. Each of these objects overlies the food with which it appeared in training. So we simply record the monkey's choices. On a separate day, we pre-feed the animal one of these two foods to satiety with the aim of devaluing that food, bring the animals to the test apparatus, and again, give them a series of probe trials, record their choices, and on a separate day, we pre-feed the animal the other food, bring them to the test apparatus, and so on. So from these data, we can collect a nice quantitative measure. Intact monkeys do a clever thing. If you pre-feed them, say, on peanuts, by the time they come to the choice test, they'll avoid the objects overlying peanuts in favor of the remaining objects, and we can calculate from that a difference score or a proportion shifted. It may appear on some slides. And that's what you'll see on the next slide. So here are some data from this kind of task. So our difference scoring on the ordinate. The open bars, or white bars, are unoperated controls. And here, the higher the bar, the better the job monkeys are doing shifting their choices, and the greater the sensitivity to this change in food value. And so this is the same color code as before. Now you can see that monkeys with selective excitotoxic lesions are impaired on this task. Interestingly, the magnitude of the impairment is the same as after aspiration lesions. So there are two things. One is it's clear here that now the neurons in the orbital cortex really do seem to be mediating this behavior, and the lesions are effective. So that's very helpful. And then the question is, okay, what does this mean? What do these data tell us? Well, I think it's telling us, at the very least, that your orbital cortex is helping you link environmental stimuli, in this case objects, with the food value, the value of the food they predict. What I didn't emphasize is, at the time of these protests, there are different objects on every trial. So there's actually a whole series of trials with different objects on every trial. There is no opportunity to relearn the association between an object and the now devalued food. The monkeys are making choices on the fly, as it were. So this means that the orbital cortex is providing a mechanism for animals to sort of automatically link the object information to the changed value. This doesn't seem like much, but it means that the orbital cortex is helping you make this connection, and without having to learn through additional errors how to make a better choice. I should also add, this is, at least by some accounts, an important aspect of goal-directed learning and goal-directed behavior. This is sort of the acid test for goal-directed behavior. By doing this, the monkeys are showing us they care about the value of the food. And by this time, you might be wondering, okay, there are all these other potential interpretations. Because these data are published, I'm not going to go through all the details, but I can tell you the monkeys, the operated groups, have intact visual perceptual abilities. They learn visual discriminations just fine at the same rate as controls. Monkeys with these lesions will work just as hard to earn food rewards as intact monkeys in a progressive ratio task. Their food preferences do not change. And finally, as far as we can tell, their satiety mechanisms are normal. So our monkeys that are going through the satiety eat the same amounts as intact monkeys. They sate in what seems to be a normal manner. And importantly, if in all our monkeys, operated and unoperated alike, if you give them a choice between the foods themselves, a visual choice between a sated food and an unsated food, all the monkeys avoid the sated food. So they can discriminate that. They tell us they don't want the food. It means that this impairment is very specific. It's just this linkage between this one more abstract level, the object information and the food value. And just because, sometimes I think it's hard to think about, well, so what does this do for an animal in the wild? I've inserted this slide. This is a new slide that I found on the internet. It just shows a macaque monkey searching under lily pads for some food resource, but that's an example of the kind of thing that would be useful for, this mechanism would be useful for, that the animal actually has to find one kind of object in the environment or one thing in the environment that's connected to abstractly food resources and the value of those resources. The devaluation task has recently been carried out in humans. This is work done in Dan Tranel's lab. I just think it's fascinating. They did a really good job setting up this task in humans. So these humans happen to have damage to large swaths of their orbital cortex, as shown here, and the medial frontal cortex, shown here. These are classically called lesions of ventromedial prefrontal cortex, but that means very different things to different people. So I just want to make it clear that these patients indeed have damage to both orbital and medial cortex. They were trained on a task that was very much like the monkeys had. That is, different stimuli were assigned to different foods. I won't give you all the details. And then they calculated a different score, in this case a devaluation score, again, very similar to the way we did in monkeys. So what you can see is the patients with this damage to orbital and medial sectors of frontal cortex were impaired in their choices relative to normal and brain damaged control groups. What's astounding is, as shown here in this little summary slide, I think, the patients gave normal ratings of these hedonic values of the foods. So just like monkeys, they could tell you, yeah, I want this food or not this food. And the patients reported a decrease in pleasantness ratings for the sated food. And then finally, the patients, as you saw on the last slide, the patients with ventromedial prefrontal cortex damage failed to reduce their instrumental choices for a food they themselves no longer deemed valuable in self-report. And I just think that those data look so much map on to those findings in monkeys that, and of course the bonus here is that these subjects can verbalize and give their self-report of no, they don't want this food, they don't like the food, but nevertheless they can't stop making these actions and choosing. So it suggests a disconnection of knowledge and action, sort of like a goal neglect. We've also tried to look at, of course, the orbital frontal cortex isn't working on its own. We've tried to identify the circuitry that is working together with the orbital frontal cortex, and from disconnection experiments where we literally remove one structure in one hemisphere and another structure in another hemisphere, which tells us, which removes the ability of those structures to interact. We've found that the orbital cortex works together with the amygdala. It's probably the basolateral amygdala, but we haven't tested that specifically. And the medial portion of the medial dorsal nucleus of the thalamus. And of course, all these regions are interconnected. So at least that's, so far, the larger circuitry that's involved in mediating this kind of behavior. So this suggests that this idea about orbital cortex updating outcome valuations seems to be correct, and literally that the idea is that the orbital cortex is representing outcomes in terms of foods and fluids, including their sensory properties and their magnitude and their value. And this brings us to the last idea. This idea about credit assignment has gained some traction recently because work from Mark Walton and Tim Behrens and Matthew Rushworth's lab found that monkeys with aspiration lesions of the orbital cortex were impaired in a probabilistic learning task. And they went on to carry out an interesting analysis to get at contingent learning and credit assignment, and found that those animals were impaired. So we're going to talk about that for just a minute. But again, the idea is that credit assignment, these all obviously are flavors of stimulus reward learning in some way. And the idea with credit assignment is that it's something more specific about assigning, as I said, a particular outcome to a particular choice. So this is the task used by Walton et al. It's really a three-choice probabilistic learning task, also called the three-arm bandit task. On this task, on each trial, monkeys see 3 images on a monitor, and the monkey's job is to select and touch one of those images on a touch-sensitive screen for the chance of earning a food reward. And these 3 images could be called choice options A, B, and C. Unbeknownst to the monkeys, each of these choice options was assigned a reward schedule that, and you can see it's changing reward schedules across a 300 trial session. So you can see that option A, shown in blue, is the best choice at the beginning of the session for about the first half of the session. But even if you choose it, you're only going to get reward on about 60% of the trials. Option C, shown in red, is the worst option. You're only going to get, well, you're getting 0 reward, literally 0. if you choose it. But later in the session, option C is the best option, and option B is the worst option. So to perform well in the task, the monkeys have to, again, discriminate the objects, which is trivial. They need to sample the different images, and then they need to track these changing reward probabilities. So, as I said, Walton et al. Found that monkeys with aspiration lesions were impaired on this task, and importantly, they interpreted the impairment as one of valuation updating, and that left us with these two ideas about valuation updating. In the devaluation task, when we talked about valuation updating, we literally meant something about the change in value of the food that's occurring during selective satiation. But in the three-arm bandit task, when Walton et al. Talked about valuation updating, they were talking about something like the associative weights between a stimulus and a single reward. It had to do, which is a very different thing. It has something to do with the relationship or the probability. So this left us with these two ideas, and the question was, well, are these potentially one and the same mechanism? And they're both mediated by orbital cortex. Can we sort that out? So one way forward was to test our monkeys with excitotoxic lesions of the orbital cortex on the three-arm bandit task, and that's what we did. At the time, Pete Rudebeck was a postdoc in my lab, and Pete was actually a co-author on the Walton et al. Study, so this was sort of a no-brainer. This is going to be a little movie to show you a monkey performing this three-choice task. So if you've ever seen monkeys, they love touchscreens. They're just like they're very fast. And I think I do this here. Maybe not. There we go. So, sorry, I should say the video was recorded at the end of the experiment, and this is an easy version of the task. The monkey is seated in a little plastic chair, so he's seated comfortably. We call it a chair. It's more like a box. So he's seated in the box inside a test cubicle, and I don't know if there's a way to turn the lights down, but okay, so you're going to hear a little plink. That's a reward being delivered, that plink. No reward. Yep. No reward. So great, thanks for that. So, really, all I can say is monkeys work with touchscreens very nicely. They're great. They're just, anyway, there are lots of stories I could tell, but now's not the time. I'll try to speed through the talk. So these are the data from Walton et al. Using this task with monkeys. And what you're seeing is, first of all, the colored dots are showing the identity and the probability of reward if you choose the best option. And then the black line and the gray shaded area are the mean and standard error of the mean are the controls. And the red line and the red shaded area is the same for monkeys with aspiration lesions of the orbital cortex. So you can see at the beginning of the session, all the monkeys are doing pretty well, actually. And I think that's because there's only one good option. Later in the session, when there are multiple good options, you can see the monkeys with aspiration lesions of the orbital cortex are impaired. And in fact, they're choosing chance, in that last half of the session. So these monkeys are definitely struggling with this task. These are the data from our monkeys with excitotoxic lesions of the orbital cortex, shown in green. And I think you can see the green is just totally overlapping the gray. So these monkeys are not impaired. And it is of interest that the good news is that the monkeys, the macaques in Oxford were performing a lot like the macaques in Bethesda. You can see they're learning these things, but there's always, when there's a change in the reward probabilities, there's sort of a lag in the monkeys responding to that. Walton et al. Also did something else. They didn't stop at just looking at monkeys' ability to track these changing reward probabilities. They carried out this interesting analysis to get at contingent learning. And what they did was, sorry. You can just push me out of the way, it's okay. What they did was multiple logistic regressions to try to understand the relationship between past rewards on the top here and past choices and their influence on current choice, which would be up here. So I should say that this kind of analysis requires gobs of data. And in fact, I just showed you one reward schedule, but the monkeys had at least 4 different reward schedules that they were tested on. So they're putting together data from several different reward schedules to get at this. And so the idea is that higher beta weights are shown with the brightest pixels. And if monkeys are doing good contingent learning, it means that if they, for example, made a choice 3 trials ago and received a reward for that choice 3 trials ago, that should be influencing their behavior out here. And again, the idea is that the more recent choices should have a bigger effect, and so the brighter pixels. So at the top are the data, again, from the Walton et al. Study. I know you can't see this very well, but on the left are the unoperated controls. And indeed, the brightest pixels are on the diagonal, and the brightest pixels are the most recent choices. On the right are the data from monkeys with the aspiration lesions of orbital cortex. And here the brightest pixels are not on the diagonal. They're actually a bit off. And those beta weights are plotted out here. So I think you can see a group difference there. In our monkeys with excitotoxic lesions, again, controls on the left, unoperated controls, it looks reasonable. The greatest correlation, if you will, is on the diagonal. And again, for our monkeys with excitotoxic lesions, they're showing good contingent learning. And again, you can see there's no group difference here. So now it looks like, just as was the case for reversal learning, it looks as though for this task that it may be that the performance on this task is disrupted by inadvertent damage to fibers of passage. So, so that suggests that credit assignment is not, at least not as tested this way, is not a function of the orbital frontal cortex. So of course we didn't want to leave it there with this negative finding. That's not very helpful sometimes. So we wanted to try to understand what frontal region might be contributing to this. So our best guess was this ventrolateral prefrontal cortex, often called area 12, 1247. And the reasons for this are that in macaques, lesions of this area actually do produce impairments in reversal learning. They're not so strong as the ones after orbital frontal cortex lesions, but they're there. And in addition, in humans, there's many studies now, and functional imaging studies, showing activations in this ventral prefrontal region associated with reversal learning. So again, they're related to reversal learning. And importantly, this ventrolateral region is adjacent to, but does not overlap with, our orbital cortex lesion. So we took monkeys from the main experiment, controls, and we gave them bilateral selective excitotoxic lesions of the ventrolateral prefrontal cortex and retested them on the 3-arm bandit task. And one more reason for thinking about the ventrolateral cortex, I'm just throwing in here, is I think you can see in this bright field photomicrograph, there are fibers in the uncinate fascicle coming over here and crossing over the lateral orbital sulcus, which I mentioned earlier is actually the boundary of our lesion, the fundus of this lateral orbital cortex. And you can see it's at least certainly plausible that fibers running in the uncinate fascicle headed for this ventrolateral cortex might be disrupted. So here are the data. On the left are, of course, the data I already showed you. On the right are our new data from our monkeys with ventrolateral prefrontal cortex lesions. I'm just going to cut to the chase. I think you can see at a glance that the scores of these monkeys, shown in blue, look a lot like the monkeys with aspiration lesions of the orbital cortex, not so much like the monkeys with excitotoxic lesions. So we think at least one answer is that the aspiration lesions of the orbital cortex may be disrupting fibers going into or out of ventrolateral prefrontal cortex. This group also has severely impaired contingent learning and that contingent learning analysis. So at this point, a key question was how do those monkeys perform on our devaluation task? So if we look at the scores of monkeys on the devaluation task, an analysis of variance shows a main effective group, and post hoc tests show that the orbital frontal monkeys with orbital frontal cortex lesions are severely impaired, and significantly so relative to controls and monkeys with ventrolateral prefrontal cortex lesions. And what that gives us is a clear double dissociation. So in this case, I've just tried to make that clear by plotting it on a single slide with the contingent learning measure on the left and devaluation on the right. But this is a normalized contingent learning score, so you can see monkeys with orbital frontal cortex lesions shown in green here have a score of 0, which means they're not different from controls. Monkeys with lesions of the ventrolateral prefrontal cortex are scoring much worse than that. And on our devaluation score, again, monkeys with ventrolateral cortex lesions are doing fine, but monkeys with orbital cortex lesions are doing worse. So I think what this is telling us is of these two ideas we started with, that these actually are, indeed, two different mechanisms that are depending on two different parts of the brain. In the case of the devaluation task, I think the orbital frontal cortex is essential for mediating the change in value that occurs during the selective satiation procedure, and that's indexing and helping monkeys make good choices based on something like desirability or palatability. And by contrast, in the 3-arm bandit task, I think the ventrolateral prefrontal cortex is helping monkeys register the changes in probability between an image or object and a reward, which is indexing resource availability. So clearly another way to express this is that this is a mechanism that's dependent on internal state, and this clearly is something that's dependent on external contingencies. So I think these are two really fundamental ways of dealing with different aspects of reward that are of course valuable in making choices. So we've also looked at specializations within the orbital frontal cortex. I won't spend much time on this, but I did want to point out that from the work of Carmichael and Price, they've described this region as the so-called sensory network, or sometimes the orbital network, and that's because projections into this region, the blue region in particular, include gustatory information, olfactory information, and importantly, visual information. And in the macaque cortex, area 13 is really the first place that's getting this conjunction of sensory inputs, and that could be useful for, you might think, for making choices of this type. And in our own studies, it turns out that it's this areas 11 and 13, the blue bit again, that actually seems to be essential for this devaluation and desirability choices. So to summarize what I've said so far, as I said before, I think the overarching function of the orbital cortex probably is not in behavioral inhibition or cognitive flexibility. I think the orbital cortex is helping us choose advantageously among options based on the predicted sensory properties of outcomes, including the subjective value. And importantly, I think this may have arisen the way it has because it depends on visual information and visual inputs, and primates are highly dependent on vision. It seems to hold for both objects and actions. Sarah Rhodes of my lab actually designed a devaluation task. This was a true action outcome task and meant to be specific to action outcome. And we found the same pattern of results, namely that you need your amygdala and you need your orbital frontal cortex as defined here for an action outcome task. As I said before, we know that the orbital cortex needs to interact with other brain regions to mediate this kind of behavior. And it's consistent with the neurophysiological data in macaques, where if you're recording for neurons in the orbital cortex in reward learning tasks, when monkeys are looking at images that predict different food rewards or different amounts of juice reward, while the monkeys are looking at the images, neurons in their orbital cortex are active in relation to the value of the outcome. So that's, even though they're looking at the images, the neuronal activity is independent of the visual properties of the images. They're independent, for the most part, of the upcoming actions that the animal may be doing. Those neurons are encoding the anticipated reward. I've already told you that. So again, just a word about what this might mean and why did it arise in primates. It turns out the granular orbital frontal cortex in early primates, that arose in early primates, those early primates were very small and were like sort of reaching and grasping and leaping. And they were pretty much confined to the fine branch niche. I'm only speculating here, but it's possible that this orbital frontal cortex conferred an advantage in just efficiently identifying valuable resources. In the case of the ventrolateral prefrontal cortex in anthropoid primates, this region arose, again, not alone, but actually with a suite of areas. And there's really not much information about exactly how this happened, but the ventrolateral prefrontal cortex probably arose about the same time as many other parts of frontal cortex, like dorsolateral prefrontal cortex, parietal regions, and there was also an expansion of visual cortex around roughly the same time. These animals, the anthropoids, were much larger primates with larger ranges, foraging ranges. So the idea is that having a mechanism, actually I put it on the next slide, but having this mechanism to identify resources at a distance may have helped them. Again, it doesn't take much to be an advantage or to be a selective advantage. And the idea is that anthropoid primates learned something like context, goal, action, outcome conjunctions rapidly and can use that information, again, to generate goals for action. Some of these ideas from Passingham and Wise, that the frontal cortex allows experience from a single trial to influence behavior, so that even though there's a lot of frontal cortex in our brains, in the end, the selective advantage may have been simply reducing errors. You just need to reduce a few errors to make something an advantage. And we've written about this at book length. So with Steve Wise and Kim Graham, who's at Cardiff University, and is a human neuropsychologist and neuroscientist, what we've tried to say is that as a series of our direct ancestors surmounted the problems and exploited the opportunities of their time and place, the brains developed specializations, new forms of representation that help them gain an adaptive advantage. And the idea is that selection acts on these representation as it does on other features, for example, toes or teeth, and that through inheritance, these representations in modified form made the cerebral cortex what it is today in all the different lineages. So our take-home messages are, and I do have a few add-ons, as I said, another section. Oh, but there's not time. Okay, that's good. So outcome is not a monolithic concept. I doubt that anybody here thought it was, but, and there are many other aspects of reward that might be evaluated, but certainly we need to take into account desirability versus availability. The idea is that the prefrontal cortex areas house different types of conjunctive representations that together guide outcome-directed behavior, and that it's all about representations. So in some sense, this is, important ideas about the frontal cortex is that it's important for executive function. But I think what we're saying is, well, it's actually not about executive function, it's about the kinds of information that are stored in frontal cortex. And that together, the prefrontal areas represent goals for action and the means to achieve them, which is an idea that's been around for a while. Recently there's been a lot of press concerning the role of orbital frontal cortex in representing state space or in representing a cognitive map. I think our findings in monkeys undermine that idea to some extent, since we've found different areas outside frontal cortex that are important for this phenomenon. And I think my sense is in monkeys that it's the entire prefrontal cortex that's contributing to a cognitive map, if you will, or to something like a state space. So current directions, I haven't allowed enough time, which is fine. So I'm just going to skip over this part and go to my acknowledgements. Okay, I'm going to go through this, which are here. So really, Pete Rudebeck, who's now at Mount Sinai School of Medicine in New York, and Alicia Izquierdo, who's at UCLA, contributed to a lot of the data to this work. And many of the ideas that I talked about were derived from discussions with Kim Graham, Steve Wise. And thank you for your attention.